Hi there. Do you know how the medicine you use for diseases work? And do you know what are the new advances in the field? Medicines, as the human body, are composed of molecules, which are a combination of atoms, the fundamental building blocks of matter. In the case of our body, more molecules combine to form cells, more cells, more tissues, organs, and more organs and organs. Thus, our cells. Now, in order to live, each of our cells uses the information stored in the molecule known as DNA, which is transcribed to RNA first, and then the RNA is converted to other molecules, the proteins, which are the effective tools employed by our cells to live. You can thus imagine your cells as a series of factories, where all the proteins are employed to allow life itself. How? As work as an assembly line, they process the molecules that they encounter, one after another, and that are needed for the life of the cell. So, every protein is important. Otherwise, the assembly line stops or does not work properly, and thus the cell, and then the organs, and then eventually our cells. Now, when a protein is malfunctioning, it's like dealing with a broken tool. Some molecules cannot be processed, thus there is missing of required functions or products or accumulation of unrequired ones, which leads to malfunctioning cells, in turn malfunctioning organs, and thus the clinical symptoms of the disease. It is as if our body is trying to drive a broken car. Some time ago, however, thanks to the work of brilliant minds and the advancement of science, the study of protein has led to the discovery of some areas on them which essentially act like locks, to which well-fitting molecules, like keys, can bind and engage or kill the activity of the protein, even when malfunctioning. The discovery of multiple locks, each bound by specific keys or molecules, has shown that these locks can influence each other. When a key binds to one lock, it can induce changes in another lock, making it easier or harder for that lock to be opened or closed by keys able to bind to it. This process, called Allostery, can lead to significant therapeutic effects. This is what me and my colleagues of the network are exploring in Allo. The work scientists like me do specifically consists in the creation, testing, and refining of molecules, which can make up keys able to bind to the malfunctioning protein in order to stop the disease, and so create the medicine we use. In particular, my contribution is on trying to optimize even more the fitting key by means of some other molecules, which as superglue are able to convert the whole key into something we can say stickier and thus long-lasting. This type of stickier key is called covalent, and when it meets its protein target, it can trap it, securing a prolonged cure, so that the disease may be cured possibly even more easily. My contribution as early-stage researcher in Allo focuses on the use of covalent keys in Allostery to target disease-related proteins, including HDAC8 and SHP2, with the aim of more effectively fighting cancer and expand the potentially pro-therapeutic application of these keys.